All right, this is take 2 of video 21 on the sarcomere. My video disappeared, so you may or may not be able to see me. But anyways, let's get rolling with video 21. There, I reappeared. So when we're talking about the sarcomere, what we're getting at is sections of a muscle at the microscopic level. So first, before we get into the sarcomere, we need to kind of whittle ourselves down to that level, because muscles are composed of lots and lots of bundles within bundles within bundles. At the very, very center, you're going to have a bundle of myofilaments making a myofibril. Now, the sarcomere is going to be at that myofilament level. So that is where we have to build. We're going to build up to a whole muscle, and then we're going to take a look at those myofilaments. A bundle of myofibrils is going to give us a muscle fiber. That muscle fiber is going to be covered with connective tissue known as the endomysium, meaning that innermost connective tissue layer. Muscle fibers, especially when we talk about skeletal muscles, are the same thing as a single cell or a single muscle cell. The endomysium is going to be really important for those muscle fibers because it's what is supplying those muscle cells with fluid, with the nutrients that they need to survive. A bundle of muscle fibers is going to make a fascicle, which is going to be wrapped in connective tissue known as the perimysium. The fascicles are going to be really important for your nervous system to be able to activate certain parts of a muscle. So you don't end up activating and contracting a full muscle and getting the full tension out of that muscle when all you need is very, very little force to be able to lift that feather off the ground. A bundle of fascicles is going to give us a full muscle wrapped in an epimysium. The epimysium is what separates the muscle from other organs, muscles, tissues, or whatever it is around it. It allows that muscle to stay intact while contracting so that structural integrity can be maintained. The three mesia, the endo, peri, and epimysium, are going to intertwine. And eventually, at the ends of the muscle, they're going to insert into tendons, aponeuroses, fascia, whatever it might be, in order to attach that muscle to whatever that muscle is pulling on. So here we can see that breakdown all the way from myofibril, the muscle fiber wrapped in endomysium, the fascicle wrapped in perimysium, and the muscle wrapped in epimysium, all coming together into a tendon in this case. Some other important terms, like I mentioned, skeletal muscle cells mean the same thing as a muscle fiber because they have a very long cylindrical shape. And in fact, they can be so long that, in one example, the muscle cells, muscle fibers in your sartorius muscle can be up to a foot long. Some other terms, the cell membrane of a skeletal muscle cell is going to be known as the sarcolemma. So that's just that outermost membrane where we have the fluid mosaic model, things moving in and out of the muscle cell. The cytoplasm is going to be known as the sarcoplasm. Remember, that's kind of the liquid space inside the cell. And your smooth ER is going to be known as the sarcoplasmic reticulum in these cells. Remember that smooth ER is one of the organelles, membrane-bound organelles, so that is called abbreviated the SR in muscle fibers. So now we can get into the sarcomere. The sarcomere is the functional unit of a muscle fiber. Basically, all contraction can be traced back to the sarcomere. If you get any smaller than a sarcomere, you start to lose important aspects of contraction. There's lots and lots of myofilaments. You have the myosin, which is a very thick protein, actin, which is a very thin protein, and then troponin and tropomyosin, which are regulatory proteins that are going to prevent actin and myosin from binding to one another, because troponin and tropomyosin is going to block binding sites on actin, which you can see down as the green and orange on this picture here. Myosin, the thick protein, they do have these myosin heads that are going to be important for contraction. You can kind of see the thick versus thin nature of the myosin and actin on this image here. The arrangement of myosin and actin, the thick and thin protein, is what gives striations in skeletal muscle. So the thick protein myosin is going to create a very dark band. Not a lot of light can pass through areas where myosin is, but actin as a thin protein is going to be a very light colored band. And so what you'll see, striations, that stripiness is alternating dark and light bands traveling all the way through that skeletal muscle fiber. So the sarcomere is going to run from Z line to Z line. We'll see some pictures of that here in a second. Z line is going to be the center of the I band. I band is going to be the light area. So remember, this is where only actin is located in the I band because that is the light area. It's the reason you see the light part of the striations. The dark area is going to be the A band. This is going to be where myosin is. Now, some myosin and actin do overlap. So when you're determining the dark area versus the light area, A band versus I band, there can only be actin. And in A band, there has to be myosin, but there may be actin as well. So it's all about whether or not myosin is there, is really what makes the difference between the light and the dark band. So you can see here the Z disc, as they call it, means the same thing as the Z line, is kind of what actin is anchored to on the edges here. And actin is going to be pulled by myosin when we take a look at in the next video. But actin gets pulled by myosin. You can see these dark areas here, the A band stretching all the way across where the purple myosin is here. It's purple in this instance. The A band where there is only actin. There is no myosin in this I band. Within the A band, you're going to have the H zone and M line. The H zone is going to be where myosin is present, but there is no actin. The H zone actually is going to change in size when a muscle is contracting. M line is simply going to be the middle of the sarcomere. Generally, it's going to be where there is only myosin present, right down the middle of the myosin, and it's going to be halfway between that Z line to Z line distance that marks the edges of the sarcomere. Here you can see that H zone denoted across the middle where there's only myosin, no actin, and the M line would sit right in the middle here. So also included attached to muscle fibers and stimulating sarcomeres are neuromuscular junctions. These are going to be areas where an axon terminal of a motor neuron is going to attach to that muscle fiber. Now, motor neurons are simply going to be neurons that are responsible for stimulating muscle movement or motor movement, some sort of movement associated with it. Axon terminals are going to be the ends of a neuron that are delivering a message. Neurons, when we learn about this during our nervous system, have a receiving end and a delivering end. Axon terminals are the delivery end. They are sending some sort of message onto whatever comes next, be it a neuron, another neuron, or a muscle fiber in this case. One motor neuron can branch and attach to many, many muscle fibers. When we take a look at a single motor neuron and all of the muscle fibers that it stimulates, that is called a motor unit. Motor units will become important later on when we start looking at contractions of muscles. So here you can see this axon terminal and how it is attached to the not attached to, but embedded into the sarcolemma, that cell membrane of a muscle fiber, and how it is going to be stimulating our sarcomere that we have sitting down here. So if you have any questions on sarcomeres, please let me know. Things are going to get very chemically oriented and very abstract, so it can be kind of a difficult topic to follow. So make sure you're asking questions either on the updates tab or in class.